drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about one of my cool new affiliate partners called Seed Oil Scout. Seed Oil Scout has a really cool map feature that helps you find restaurants in your area where you can find seed oil-free options. They also have a really cool product scanner, so you can take out the app and use the camera functionality of your phone to scan products in grocery stores and see what's actually in these things. Do they contain seed oils? Do they contain other things that you want to know about that aren't necessarily on the box? This will help you find places to eat and find foods to eat that actually have what you want in them and help you avoid those that don't. So if you're looking to cut back on your seed oil consumption, or you just want to know more about what's in the foods on the shelves at the grocery stores, download Seed Oil Scout today. If you go through the link in the episode description, click into that and you'll see a link to the Seed Oil Scout app. If you go through that link on my support page, you'll be directly helping the podcast, and you'll be able to download this really cool, easy to use app that will help you find healthier food options. So go through the link in the episode description and download Seed Oil Scout today. Visit mindandmatter.substack.com to find all of my content. Joining me.
Thank you for the invitation, Nick.
Do you want to start off by just telling everyone a little bit about who you are and what your lab studies?
I'm Gero Miesenböck. I'm a professor of physiology at the University of Oxford in England. We work on neurobiology in general, and more recently, specifically on the neuronal control and biological function of sleep.
And what sort of creatures do you study?
Fruit flies and increasingly also mice, because we want to be taken seriously.
What do you mean by that? And so why do you and why do a lot of scientists study flies? What is, for those that are not familiar with experimental research, fruit flies are very common, and sometimes when you're not familiar with that, that's puzzling at first. Why would you be interested in fruit flies?
They have compact brains that are capable of quite remarkable cognitive feats. As Francis Crick and Seymour Benzer debated the relative merits of model organisms, Benzer always said to Crick, they can do everything you can do, Francis, plus they can fly and land upside down on the ceiling, something you can't do. So there's a behavioral richness there that's controlled by a very compact nervous system, that we have powerful genetic tools developed over more than a century that we can use to dissect it. And more recently also a complete electron microscopy derived synaptic resolution wiring diagram. So it's a really, really, really fertile experimental system. But in many cases, and this is why I only half-jokingly said we want to be taken seriously, people want to see that what you discover in an invertebrate is also relevant to the mammalian and especially the human brain. So we need to show that there's generality to the kinds of things that we unravel.
Mm-hmm. And in many cases there are, because at the end of the day, even though a fly is much smaller and simpler, they have many fewer neurons and so forth. They do, fundamentally have a lot in common with a mammalian brain. They're built out of the same components, and they do a lot of the same things. And so there are, and I think part of what we'll talk about today are some of the fundamentals that are the fundamental things that are happening in both vertebrate and invertebrate brains.
Yes, you're preaching to the choir.
And so a lot of today we'll talk about your work to study sleep and what is actually going on under the hood in brains and nervous systems to perform sleep, and why sleep needs to happen. So fruit flies sleep?
They do.
Yeah.
It's been known for about a quarter of a century. There were two papers published in 2000, one by Amita Segal's lab and the other one by Giulio Tononi and Chiara Girelli's lab, that showed for the first time that rest in flies is a sleep-like state, and then from that a lot of fundamental mechanistic work has evolved and derived, including our own.
Mm-hmm. And so, do all animals seem to sleep where we've looked for it? And does that tell us that there's something fundamental that's there and present in the nervous system, even of the simplest animals with the simplest nervous systems, that make sleep necessary for the brain?
As far as we can tell, yes. In fact, you don't even need a nervous system to require sleep. There was a discovery reported by Leah John Toro at Caltech in 2017 that even jellyfish, which don't have a centralized brain, they just have a distributed nerve net, show a sleep-like state. Okay, and that, I think, that observation tells you something very, very fundamental. As you know, I mean, sleep is the only ubiquitous behavior whose function we don't understand, and whose function we can't infer simply by observing the sleeping animal, right? I mean, in contrast to, say, eating or drinking, where it's obvious what the action is good for, sleep doesn't really give you a clue if you just observe a sleeping animal. So there's a huge variety of theories of what sleep might be good for, including sophisticated cognitive functions such as hippocampal replay of newly acquired memories and so forth. I would not argue that these functions don't exist, but I would argue that they can't be the primary ancient evolutionary driver why sleep was necessary if a jellyfish already requires sleep. And it obviously doesn't have a hippocampus, and I'm not sure what its cognitive abilities are. So I think there has to be something really, really, really basic.
Yeah. So there has to be something basic, because all of these creatures need to sleep, even creatures that don't have to do spatial navigation like a mouse or even a fly can do. And so basically what you're saying, and I think what we'll kind of unpack today, is there must be some very fundamental things happening that probably have to do with cells and metabolism and stuff that even a jellyfish needs to take care of. And then there are functions of sleep related to things like memory consolidation and synaptic homeostasis and all of this stuff. But these are probably things that came later and got stacked on top of this more basic and fundamental and ancient function.
That's exactly how I think about the problem. I mean, to let the cat out of the bag, I think that sleep has an ancient metabolic origin. It's specifically tied to respiration, the enormous energy gains that you have when you use mitochondria to transfer nutrient-derived electrons to oxygen as the terminal acceptor. That process has the advantage of maximizing the free energy yield of your food intake, but it has side effects, and we think those side effects require sleep to offset them. Mm-hmm. But once we think evolution has sort of come up with that solution of putting animals to sleep for a few hours, then of course these off periods were used for other things, such as synaptic homeostasis or memory consolidation or other kinds of things. So our hypothesis is inclusive rather than exclusive.
Right. Yeah, and that's kind of a common theme in evolution. If something evolves, an adaptation or a mechanism gets put in place and it needs to be there, very often it will get tinkered with or co-opted into a new thing, so that you preserve the old function and then you sort of start reusing it and retooling it for other things that come later.
Exactly, especially if that function has significant risks and costs, as sleep does. I mean, the famous sleep researcher and one of the founding members of the Chicago School of Sleep Research, Al Rechtschaffen, he quipped that if sleep didn't have an absolutely fundamental purpose, it would be the biggest mistake the evolutionary process ever made, because of its risks and costs. Right. So there must be some really important benefit that offsets these risks and costs, but we don't really know what that benefit is.
And of course, we all sleep every day or almost every day most of the time. And when we don't, we feel it. So we have an intuitive understanding that there's a recuperative function to sleep. Clearly it has something to do with energy. It affects your memory, it affects your brain function. But the devil's in the details here. What exactly is going on there? That's what's been mysterious. So when we start thinking about the recuperative function of sleep, you mentioned some of the theories that have been out there. You sort of mentioned the punchline in essence of what you think is going on. This has to do with oxygen-based respiration and mitochondrial metabolism. So what we get from having mitochondria and needing oxygen to generate energy is we get a lot of energy. We make a lot of ATP with our mitochondria, but you said there's side effects. I want to give people a sense for the basics of the electron transport chain that's in the mitochondria and why that gives us so much energy, but also how the sort of side effect side of this works. So at a very high basic level, we don't need to be comprehensive here. What is the electron transport chain in mitochondria, and how does oxygen fit into energy production? Why are we breathing this oxygen?
So you can think of the electron transport chain in the inner mitochondrial membrane as just a wire that conducts electricity. Except the wire is made out of proteins, large protein complexes. There's 4 of them that make up the electron transport chain, and they are encoded by 2 genomes, by the nuclear genome and also certain subunits, and strikingly, the ones that are most intimately involved with electron transfer are encoded by the mitochondrial genome. So these 2 genomes have to somehow interact with angstrom scale precision and build this wire that then conducts electrons. It's essentially, it passes a current from complex 1 down to complex 4, where the electrons are transferred to oxygen. Now, if you look at the redox potential difference that you can tap through this transfer, it's large. It's more than a volt. This is why oxygen tanks have danger labels, right? It's a very explosive reaction that biology has learned to tame by breaking it down into 3 steps. So you have 3 transfers of the electrons, first from NADH, which comes out of the Krebs cycle in the mitochondrial matrix, to complex 1, then to complex 3, to complex 4, and to oxygen. So you release the huge amount of energy in a gradual piecemeal fashion. These electron transport complexes, or the current that flows through that wire, then enables proton pumping by these electron transport complexes. There's a proton gradient that's stored as in a battery across the inner mitochondrial membranes. And then the protons flow through complex 5, which is the ATP synthase, which is a nanoscopic turbine that spins in the inner mitochondrial membrane, and that makes ATP. Now, the wonderful way of doing this chemiosmotic coupling, the combination of electricity that's central to ATP synthesis, is that it frees you from the shackles of stoichiometry. If you did a chemical group transfer, you would always have integral reaction partner ratios, right? But by interposing an electrochemical gradient, you can actually squeeze out the last fraction of energy that you can need, because the stoichiometry doesn't have to be 1-to-1 or 2-to-1 or 3-to-1.
Right.
Or what. Right. Okay.
Right. If we were doing things strictly in a chemical fashion, you would need a fixed number of ultimately particles from food to make 1 unit of energy. But because we have this flow of electrons creating this proton gradient, and it's flowing through this little molecular turbine, it's just a much more efficient way to generate as much molecular energy as you possibly can, basically.
Exactly. And the availability of oxygen to act as the terminal electron acceptor then really increased the amount of energy that you could squeeze out a lot. And that's something that happened in Earth's evolution around 2.5 billion years ago, because life, it's thought, evolved on an anoxic planet. There was no oxygen in the atmosphere. All the oxygen that's around was actually generated by life. Mm-hmm. And oxygen levels sort of rose gradually, but then with two large stepwise increases. The first one about 2.5 billion years ago, and that's the time it was thought that then triggered the symbiosis between two forms of bacteria that gave rise to eukaryotes containing mitochondria. Mm-hmm. And the second large increase in atmospheric oxygen levels was about 570, 580 million years ago. And that's when multicellular life really exploded, because you could just gain so much more energy. And complex multicellular life became possible. So this some people think was the spark that lit the Cambrian explosion of multicellular life. Mm-hmm. The appearance of nervous systems or neuronal nets, and the appearance of sleep. Okay. Mm-hmm. So we've talked about all the advantages that electron transport and oxygen has. What's the downside? The problem is the electron transport chain in the mitochondria uses single unpaired electrons, okay, which is unusual for a redox reaction and something that oxygen loves, because oxygen is a very peculiar gas. It has in its outer shell two orbitals that have one electron each, one unpaired electron, and those spins of these two electrons point in the same direction. That means that if you want to engage oxygen in an electron transfer reaction, you need to add the electrons one at a time. Mm. Okay. So this is something that in a normal redox reaction, which always transfers electron pairs, that doesn't happen. So that has advantages. For example, the so-called spin restriction on the reactivity of oxygen prevents the spontaneous combustion of the atmosphere, which is a good thing. Mm. But it means that if you have unpaired electrons somewhere, such as in the mitochondrial electron transport chain, their chance of leaking to oxygen is very high. Oxygen is like the magnet for these things. So basically, the wire of the electron transport chain becomes poorly insulated in the presence of oxygen. Once such an electron leak in the mitochondria happens, you form something called reactive oxygen species. So a molecular oxygen that has only a single unpaired electron. And then we think that is a very, very dangerous thing to happen.
Mm-hmm. So, basically, you want this electrical current flowing because it's enabling all of this amazing metabolic stuff to happen. But just like, I have electrical wires in the home that make, the lights and the TV and all of that possible. You want this to be well insulated. You want everything to be protected, and you don't want the electricity leaking and sparking and lighting your house on fire.
Exactly. And the leakiness also seems to be determined to some extent by how well the mitochondrial and the nuclear encoded electron transport chain proteins cooperate together. So every individual is different in that respect, because the mitochondria only come from the mom, and the nuclear genome obviously from both mom and dad. So every individual is a new experiment that has to find out how these two components actually work together. And it's even thought, I read somewhere, that one of the most common reasons of spontaneous abortions in humans is actually mitonuclear conflict. So very early during embryonic development, the match, the mitonuclear match is checked, and if the match is poor and you leak a lot of electrons, the developing embryo at a very, very early stage undergoes apoptosis.
Yeah. So depending on who mom and dad were, the amount of leakage of these electrons can be greater or lesser. But in everyone, to some extent, there's always, it's never perfect. And there's always going to be some leakiness. There's going to be some reactive oxygen species. And that is an inevitable consequence of this oxygen-based metabolism.
Exactly. That's what we think is what's going on. And that's what we also think is the root cause of the need for sleep. In animals that have neurons that are energy or power hungry.
Yeah. So, I mean, so this makes a lot of sense so far. If sleep is fundamental, meaning that we see it in everything from jellyfish to flies to mice to humans, it has to be serving some function that's also fundamental. And jellyfish don't have a hippocampus. Neither do flies, for that matter. There has to be something that all of these animals need, and what they all have in common is this metabolism, this oxygen-based metabolism that uses mitochondria to create energy.
Exactly.
And so let's talk a little bit more about the side effects of aerobic metabolism here. So we're using oxygen as an electron acceptor. It's got advantages and disadvantages. When we have this leakiness in the mitochondria, when we generate reactive oxygen species, why is that bad? What do they do? What are they lighting on fire that creates side effects or problems for our cells?
So reactive oxygen species, as the name says, are very, very reactive molecules. They target multiple cellular constituents, from proteins to DNA to lipids, and cause either covalent modifications of proteins or lipid peroxidation. And we think that lipid peroxidation is one of the key events that happen if you have a large mitochondrial electron leak. The reason is that lipids obviously are right next to the electron transport complexes in the inner mitochondrial membrane. They are highly abundant, and they are capable of entering a chain reaction once you produce one peroxidized lipid. So if you have, say, a membrane phospholipid with a polyunsaturated fatty acyl chain, and you hit that with a reactive oxygen species, you form a lipid peroxyl radical. And that can then propagate the chain by reacting with another lipid, sort of diffusing laterally in the membrane. There's estimates that suggest that that one such hit can produce hundreds or thousands of peroxidized lipids. So the reaction can really spread like wildfire.
So literally, we're talking about lighting fat on fire, and one spark, like one oxygen radical, can light hundreds of thousands potentially of lipids on fire. And so you can imagine, literally it's like burning a hole in the wall of your house or something.
Yeah, yes, that's the way you can think about. I mean, the lipid peroxidation is usually what makes fats rancid rather than lights them on fire. But the metaphor of lighting the membranes on fire is perfectly fine.
And so the membrane, so mitochondria have two membranes. They have an outer membrane and an inner membrane. The membrane is made out of lipids largely, and those lipids can be saturated, monounsaturated, and polyunsaturated. What is the difference between those fatty acids, and what's the relationship between the saturation of the fatty acids and their ability to react with these reactive oxygen species?
So it's usually the polyunsaturated lipids. They have their chemical structure. There you have something called a bisallylic carbon atom. That's the primary target for attack by an oxygen radical, and that's how you start this lipid peroxidation chain reaction. Now, interestingly, as this chain reaction now not spreading like wildfire, but looking at the individual lipid molecule, as that progresses, the molecule undergoes a series of rearrangements and scissions that ultimately then lead to the production of carbonyls, aldehydes and ketones that are collectively also known as reactive electrophiles. So malondialdehyde is one of them, methyl glyoxal. There's lots of these reactive electrophiles that are produced in cells. Now, one of the findings that actually put us on the scent of mitochondria as a potential cause of sleep was that we had begun by studying the biophysics of specific sleep control neurons in the brains of flies. Mm-hmm. And we sought to discover how varying sleep need regulates the electrical activity of these sleep-inducing neurons. And that search zeroed in on two potassium channels, one of which contained a subunit that had sort of a mysterious structure. It looked like an enzyme whose endogenous substrate was unknown. Mm. And we found also relatively recently that the most likely endogenous substrate of that channel subunit that regulates the induction of sleep are lipid peroxidation-derived carbonyls. So you essentially have a little sensor at the plasma membrane that registers what's happening in the mitochondria, how much electron leak you have, how much lipid peroxidation you consequently have, and that is then able to pick up the accumulation of these longer-lived lipid peroxidation products over time.
So you generate a reactive oxygen species as an inevitable consequence of oxygen-based mitochondrial metabolism. They can induce things like lipid peroxidation, and they're going to produce nasty chemicals that can break stuff in the cell. You're basically saying there's a sensor, at least one sensor, that can basically keep track of how many of these nasty components you've made. And then you can immediately think of why that would be useful, right? The cell's going to want to know if you've made a lot of these things or little, or how long they've been made. And you can start to imagine how that could be hooked up to some kind of recuperative mechanism that might sort of shut things down so you can, fix things.
Exactly. So we think that, and the sensor has a very interesting property, and that is that it registers an encounter with one of these lipid-derived carbonyl molecules, then changes its state. So each of these molecules can hold one bit of information. It flips from one state to the other, and then it stays stably in that other state until it is reset by electrical activity. So you can imagine if you have one of these sleep-inducing neurons that has thousands of these channel and sensor molecules, and if you could observe the cell and these molecules over time, as the mitochondria burn fuel and respire and use oxygen, and there's sort of a gradual electron leak and gradual levels of lipid peroxidation, these carbonyls appear one after the other. And once a carbonyl hits one of these sensors, it flips its state.
So the cell can literally basically count and remember the past history of producing these nasty chemicals as a consequence of oxygen-based metabolism. And then at some point, you reach a threshold or whatever, and the neuron becomes active. And you're saying this is already a neuron that was known to induce sleep. So essentially, if we put these things together, you're saying the neurons naturally get turned on by a signal of how many of these nasty metabolic byproducts have been produced.
That's exactly what seems to be the case. So you can think of it sort of that the total amount you've spent awake, or what's called technically sleep pressure, your need to sleep, is sort of a continuous variable, right? It's sort of and you have many of these digital binary devices. And so what the cell does through this counting mechanism, it quantizes the continuous variable into the discrete binary states of this large population of ion channels. So it's like in an audio recording where you convert the analog waveform of music into the bits that you then store in digital form, right? And the cell does the same thing. It converts the analog appearance of these lipid peroxidation products into a digital memory that's then read out. And and induces sleep when the fill level of the system reaches a certain level.
And remind me, so this sensor is a channel, and is it in the mitochondrial membrane or the cell membrane?
It's in the cell membrane.
Okay.
And that also solves we were always scratching our heads before we found this crucial intermediate of lipid peroxidation products, how a signal that's short-lived, such as reactive oxygen species, could reach the electrical impulse generating machinery in the cell membrane when it originates in the mitochondria. And the answer, of course, is that it's not the short-lived reactive oxygen species that serves as a direct signal at the plasma membrane, but the lipid peroxidation products that are produced as a result of the electron leak.
Got it. So the reactive oxygen species are so ephemeral that they don't last long enough that they can kind of be detected and counted and kept track of. But these lipid peroxidation products do. So on the one hand, they're a good signal to listen to because they're nasty. They're clearly indicating that like a lot of metabolism has happened and we need to know that. But they're not so ephemeral that you can't keep track of them.
Yep.
Interesting. Okay, so and just to say it explicitly for people, normally when we think about memory, we often think about the connections between neurons and things like this. But we're talking about a system here that's all within one cell. This is one cell that's keeping track of its basically its metabolic history by converting this analog signal into a digital signal. Right.
And I mean the memory. So memory obviously has a neurobiological and a psychological sense. But it also has an engineering sense in computer science. And the memory that's in our computers and in our cell phones, known as DRAM or dynamic random access memory, that was invented in 1969 by Bob Dennard, who worked at IBM Research in the Hudson Valley. And if you look at his patent, the architecture of an elementary memory unit that can hold one bit of information precisely recapitulates the architecture of the ion channel that we've studied. So Dennard's electronic memory is a two-component system. It contains a capacitor that stores one bit in the form of an electrical charge, and then a gate, an access transistor that determines, depending on its voltage, whether you can read or write memory to the capacitor. So you can charge or discharge the capacitor. In our case, the memory unit is that ion channel subunit that flips from a reduced to an oxidized state, depending on the availability of lipid peroxidation products. And whether it can be read out is also determined by the voltage across the plasma membrane.
So human beings dreamed up and built this type of system, and they patented it, and they had no idea about this biology. And it turned out that nature had invented more or less a perfectly analogous system hundreds of millions of years ago.
Yep.
So let's go to the flies now. So we've built sort of a base here, and we'll sort of connect the dots over the next little bit of the conversation. Let's talk a little bit about flies and sleep and what was already known about the neurobiology here. So it sounded like you already knew that you have these very important sleep-inducing neurons which were sufficient to make flies sleep. You can turn them on and make flies sleep. Talk a little bit about the circuitry and the cells here that we already knew about that were really important for sleep in flies.
So the neurons that we studied are cells that project to a particular layer or two layers of the dorsal fan-shaped body in the central complex. These neurons represent a small minority. There's 30 or 31 cells among the 100,000 or so neurons that make up a fly's brain. Their role in the induction of sleep was discovered by a former postdoc of mine, Jeff Donnelly, when he was a graduate student with Paul Shaw at Washington University in St. Louis. So this was part of his thesis work. And then when Jeff came to work in my lab, we began to explore the function of these neurons and discovered that they are important for regulating sleep depending on sleep history. So they have nothing to do with the circadian clock. They are effectors of sleep pressure, and they, as such, they must therefore also be equipped to sense sleep need. So much of our work has been devoted to understanding how these neurons transduce a perceived need for sleep into sleep-inducing electrical activity.
Mhm.
I think the really important conceptual a point to make here is that it's been known for a very, very long time that sleep loss or normal sleep-wake cycles are associated with numerous changes in the brain, from electrical firing patterns to the changes in synaptic strength to metabolite concentration changes to changes in gene expression. But if you just look anywhere in the brain, it's extremely difficult logically to disentangle cause and effect.
Mhm.
If you look at these dedicated sleep-inducing cells, you know that whatever is the fundamental driver of sleep must connect in these neurons with the biophysical machinery that regulates the electrical
Right,
activity.
right.
This
You
was
you knew these neurons were important for sensing sleep need at some level, that they are sufficient to induce sleep. So whatever the natural signal is that corresponds to sleep pressure building must be sensed basically by these neurons. And what we were talking about before is getting at like what they're actually sensing.
exactly. So I think it's also a wonderful example of something that I often say. So early in my independent career, I was involved in the invention of optogenetics. And I always say that optogenetics is a beginning and not an end. So in the case of these sleep-inducing neurons, what Jeff and I did, and Jeff and Paul, was to activate these neurons artificially and see that they can induce sleep. So you can establish the causal role in the induction and maintenance of sleep. But of course, the interesting biology begins when you understand what the natural signal is that normally turns these neurons
Yeah.
on.
Yeah. And so basically the idea we're going to is, you're awake or flies awake for some time. Obviously, the longer you're awake, the longer your cells have been doing what they do, using oxygen and mitochondria to create energy. More metabolites have been made. Some of these lipid peroxides have been made. The cell is able to sense and remember these things. And at some point, this sort of builds up in the cell and causes these neurons to turn on. And then that's basically what causes sleep. When you started doing some of the work that you just published, let's talk a little bit about how you sort of zero in the fly brain on which neurons are important and where things are happening. That's going to take us to a technique that you use called transcriptomics. So what's transcriptomics, and how did you use it to figure out like where to look in the fly brain here?
So transcriptomics is a method that allows you to measure levels of gene expression genome wide, and in the version that we used, which was single cell transcriptomics, with the resolution of individual cells. So you can see which genes are turned on or off in individual cells under different conditions. So we looked at the entire central brain of flies, were able to identify specific neurons, and had flies that were either fully rested, so collected at 8 o'clock in the morning after a night of shut eye, or they had been sleep deprived the preceding night. Transcriptomics did not tell us where to look for gene expression changes. We already knew where to look based on these biophysical experiments and optogenetics experiments that I had described before. We actually used transcriptomics just to confirm in an unbiased, completely open-minded way the crucial role of mitochondria. We played ignorant.
Yeah.
We already knew from our more mechanistic work that we had this sensor in the plasma membrane, that ion channel that responded to lipid peroxidation products, and that mitochondria was sort of the source of the trigger that would spark the peroxidation reaction.
So you have these key cells in the fly brain that induce sleep. You already know that. You already know they're important. You've also independently discovered this sort of lipid peroxide sensor that we talked about before. So then you can do transcriptomics on these cells, and what that's doing is it's allowing you to look in an unbiased fashion at what genes are turned up and turned down when an animal is deprived of sleep. So you basically prevent the flies from sleeping. You can isolate these neurons that we already know are crucial for sleep, and then just look at the whole genome. And it's like, what kind of genes are up and what kind of genes are down?
Exactly. And the only genes that were up were mitochondrial.
And it didn't need to
So
be that way, right? That's kind of a striking result.
yes. So this was a nice moment when we saw that all the upregulated genes, almost without exception. I mean, all of these measurements, of course, have some level of noise. So there's other things in there. But if you try and group the sort of the genes collectively that are upregulated, the data really rub your nose in the mitochondria as being crucially important.
Yeah, and there's a lot of genes that could have been up or down regulated here. So, a relatively small fraction of the genome really pops out here, and you're saying just it all happens to be mitochondrial related genes.
Yeah.
Mostly.
Yeah.
Okay, so go ahead.
The question, of course, became, what is this? Why are the mitochondrial genes upregulated? Is this a damage response? Is this just mitochondrial proliferation? What is it? So we decided to look at the mitochondria just in these 30 or so sleep-inducing cells. So we labeled them with a fluorophore in the living flies, and then we again went through sleep deprivation versus rested, and we saw that sleep deprivation caused drastic morphological changes in the mitochondrial network. So the mitochondria became fragmented, very small. They attached to the endoplasmic reticulum, and part of the mitochondria also got taken up into lysosomes, a process called mitophagy. So it very much looked like a damage response, which is consistent again with the idea that you really exercise your mitochondria. You have a significant electron leak. The electron leak creates all the downstream problems that we've discussed. The mitochondria and their membranes get damaged as a result. And so what you do is you sequester the damaged bits, you put them into lysosomes to discard them, you attach to the endoplasmic reticulum to reload your mitochondria with lipids that are synthesized in the ER, and you upregulate mitochondrial gene expression to rebuild the organelles during recovery sleep.
Yeah. And so basically, right, the longer that mitochondria have been working or turned on, let's say, the more chances there are for them to become dysfunctional, to become damaged. They're generating these reactive oxygen species and these lipid peroxides. That's causing a problem. The longer you let that go without sort of resetting or replacing those mitochondria, the more the cell's going to be in trouble overall. And so that's basically what we're talking about here. The animals are sleep deprived. They're not allowed to sleep. The mitochondria start to become dysfunctional. They look weird. They're aggregating in different parts of the cell. And the cell has to literally digest them and replace them.
That's the idea.
Yeah. Interesting. And so we've talked about these lipid peroxides. We've talked about how polyunsaturated fatty acids are more prone to react with reactive oxygen species and produce these nasty chemicals. Talk a little bit more about the endoplasmic reticulum and how the new mitochondria get made. So it sounds like new mitochondria can get made. They kind of dock onto the ER, and this is how they get loaded with like their lipids and the stuff that they're built from.
So this is one of a really important area in modern cell biology that has grown enormously over the past 15 or so years. The study of interorganelle contact sites. So there's numerous such tightly regulated contact sites, including sites that link the mitochondria to the endoplasmic reticulum. We now even have structures in yeast that show the ER mitochondrial contact, and it's in fact a lipid channel. So you can import lipids from the ER into mitochondria. There's a hydrophobic tunnel, and you can pass the lipids through. The reason why you need this, of course, is that many lipid biosynthetic pathways are either exclusively concentrated in the ER or distributed between the ER and the mitochondria. So you have to import precursors from the ER into mitochondria. So this, again, finding the mitochondria attached to the ER suggests that something is going on with the lipids, as we've already inferred from studying the ion channel. Now, mitochondrial fission usually happens at these contact sites between mitochondria and ER. So the sequence that normally happens is the mitochondrion attaches to the ER and then recruits a dynamine-related protein that sort of constricts it and divides the organelle.
Okay, so mitochondria, they can grow in size, they can fuse together, they can split apart, and you can sort of watch and map all of this out happening in the cell, and it tells you something about the health and the state and whether or not these mitochondria have become dysfunctional.
Exactly, and there also seem to be clear relationships between mitochondrial morphology and mitochondrial function. Mm-hmm. So it's widely thought that if you have hyperfused mitochondria, so very large extended branch networks of mitochondria, that allows particularly efficient ATP synthesis, whereas if you have small, rounded, fragmented mitochondria, the efficiency of electron transport and ATP synthesis drops.
Mm-hmm. Got it. So in general, more smaller mitochondria is indication of inefficient energy production, and sort of bigger fused together mitochondria indicates the opposite, it seems.
Right. Yeah.
And so you say in this paper that mitochondrial electron surplus induces sleep. What does that mean, and how did you measure that?
So we use the mitochondrial electron surplus as an alternative expression to the electron leak that we've been talking about. So if you feed more electrons into the mitochondrial transport chain, then you need to fuel ATP production. You have a mitochondrial electron surplus, so it's like everything in life. It's a question of supply and demand, right? So we performed experiments where we either increased or decreased the supply of electrons, or increased or decreased the demand. So these were genetically targeted manipulations of the inner mitochondrial membrane, where we changed this relationship between supply and demand. Let me give you one example. We talked before about how the electron flow down that wire leads to the buildup of a proton electrochemical gradient that is then tapped by the turbine to synthesize ATP. Now there's a class of proteins called uncoupling proteins, which in fact short circuit that proton electrochemical gradients. They are little proton selective pores in the inner mitochondrial membrane. So you can imagine that if you punch a hole into the inner mitochondrial membrane, you dissipate that gradient. In order to synthesize the same amount of ATP, you need a larger supply of electrons to compensate for the hole, right? So this would be an example of reducing the mitochondrial electron surplus. And as predicted, if you overexpress such an uncoupling protein in the sleep-inducing neurons, you reduce the demand for sleep. Mm-hmm, mm-hmm. We've also done an opposite manipulation where we used a light-driven proton pump to build up the proton electrochemical gradient independent of the electrons that get fed into the transport chain. So in this way, we have sort of hypercharged the mitochondrial battery. Whatever electrons are fed in from the Krebs cycle are redundant. That means that the electron surplus increases, the electron leak increases, and sleep should increase. And that's exactly what happens.
Mm-hmm. I see. So if there's a greater electron surplus and you manipulate the system in a way that gives you that, the animals sleep, you induce sleep, basically.
Yep.
Interesting. So why? So the cell has ways to couple or uncouple electron flow to ATP synthesis. How closely those things are matched is important here. You're basically saying, you could have the current flowing through this wire That's the inner mitochondrial membrane. If the current exactly matches the amount of ATP you need, things are kind of balanced. If you have more electrons than the ATP demands of the cell, that's more electron leak. There's more chances to induce some of these metabolic byproducts that are not good for the cell. That's going to increase sleep need because sleep is going to sort of reset or recuperate the production of those things. The cell has things like uncoupling proteins, so the cell can decide and regulate how coupled the system is. Why does that exist? Why does the cell have the ability to tune and change how coupled the electron flow is to ATP synthesis?
It's not entirely clear, except in one context. So I think the best understood role for these uncoupling proteins is in thermogenesis, in the generation of body heat in brown fat. That's how these proteins were actually discovered. So the brown fat cells use these uncoupling proteins to actually turn mitochondrial electron flow into heat rather than ATP. So this is one example. In other contexts, there is evidence that uncoupling proteins play a role in the central regulation of energy metabolism in the hypothalamus, for example. But how widespread their use is in the nervous system to actually match electron supply and demand, I am unaware of any detailed studies that address that. In general, it's thought that all these forms of regulation are rather slow. So if you have a mismatch between electron supply and ATP demand, you can't correct that at the time scale of, say, action potentials, which may sort of lead us to the deep mystery that I've been obsessing about since we found that mitochondrial role, which is, okay, if the reason why we need to sleep is such a simple biochemical problem, such as electrons leaking from your mitochondria, right? Why do you need to lose consciousness to fix that problem? Why can't you find a simple biochemical solution? And I don't have an answer, but I hope to have one at some point.
And so you can artificially induce more electron leak and electron surplus. That basically causes more sleep pressure and sleep need. That makes sense. And so I guess the way to think about this in the natural state is the animal's awake. The longer it's awake for whatever reason, the more metabolism that's happened, the more excess electrons and reactive oxygen species and stuff that will have been produced, the more the lipid peroxide sensor we talked about before will be registering that all of this has happened, and the more inefficient the process will probably be as time goes on. And so these things are basically read out by the cell, and you're coupling this readout of these metabolic byproducts to the activity of special neurons and circuits in the brain that we know induce sleep when they become active in the right way.
Exactly. That's the way we picture the mechanism. Yes.
And because we're talking about mitochondrial dynamics, ATP production, this immediately, it makes it very easy, I think, for me to think about like why you would also, in other contexts, especially in so-called higher organisms, why things like memory consolidation and synaptic protein synthesis and all of these things would be correlated and tied with sleep, because those are all energy intensive processes. And obviously, if you can't reset and maintain the efficiency of the energy generation mechanism of the cell, then all of that other stuff downstream is not going to be possible either. Exactly.
And that's true not only for high organism. Flies also have serious memory deficits if you sleep deprive them. So even their memories require sleep for consolidation.
And so you think that this is probably basically why sleep evolved. This it's tied to aerobic metabolism, the use of oxygen in mitochondria. That's why you see everything that you see in flies. It implies that this stuff sort of would have to be true in vertebrates as well, because we have the same basic mitochondrial based metabolism, but also why something as seemingly simple as a jellyfish without a centralized brain would also have a sleep-like state.
Yes, that's the way I think about it.
And so
it needs to be formally shown in mammals. I mean, I think we all operate under the well-founded assumption that evolution is conservative, but you still need to demonstrate that similar mechanisms operate in the mammalian brain. And that's obviously another thing we are currently trying to do.
And so to the extent that sleep is a natural consequence of aerobic metabolism based in mitochondria, this also ties into certain patterns that we see across the tree of life, which are really interesting, and I would love to get your comments on. So one of these patterns is that smaller animals basically have faster metabolisms. They also don't live as long, and they sleep more. So when you look at, say, a mouse compared to a human, compared to a very large-bodied animal, right? The mouse doesn't live nearly as long as we do, and it also needs to sleep more hours per day than we do. Why is that, and how does this maybe tie into the oxygen part of the story here?
So the relationship between body size, mass-specific energy consumption, and sleep need was first noted by Al Rechtschaffen, who I already mentioned, in 1974. And then it was sort of formalized and I think really nailed in a paper by Van Savage and Jeffrey West of the Santa Fe Institute, which found that there's a power law that underlies this relationship. So if you measure mass-specific oxygen consumption, so oxygen consumption per gram tissue per unit time, and you plot that versus daily sleep amount on a bilogarithmic scale, you get a straight line. Now, if you look at the exponent of that straight line, it's not a multiple of 1/3 as you would expect from sort of classical Euclidean geometry, right? That it's sort of surface to volume ratio that matters. But the exponent is a multiple of 1/4. And West had done a lot of work on sort of scaling laws in various contexts and derived the idea that if you get that you get quarter power scaling based on the geometry of centralized research distribution networks, which is a fancy name for things like your cardiovascular or your respiratory system.
Yeah, effectively, it's like there's an extra dimension here beyond the 3 dimensions of space.
Exactly.
Yeah.
And it's sort of the fractal dimension of these distribution networks that underpins that quarter power scaling. So, and if you are a smaller mammal, you have a higher capillary density of these distribution networks, and the distribution networks allocate more oxygen to each of your cells per unit time.
Yeah. So a mouse can get more oxygen to more cells faster than our bodies can, than a blue whale's can.
Than a blue whale can. And that has consequences for our entire organismal physiology. So if you are small, or the smaller you are, the faster your heart beats usually, the shorter your life, and sort of tragically, the greater a fraction of your short life you spend asleep. So a mouse or a tree shrew, they sleep 18, 20, 22 hours a day. An elephant can get away with like 3-4 hours of sleep a day.
Yeah. So basically, they're in some sense they can use oxygen more readily. You say in the paper, a small animal can burn hotter, and there's certain advantages to this from like an energetic standpoint. But the trade-off is, and you say this explicitly in the paper, the price you pay for being able to burn hotter and have this sort of more efficient distribution of oxygen to your cells is a shorter life, and you spend more of it asleep.
Exactly,
because your brain has to compensate for all of the side effects of what you're actually using that oxygen for. The generation of the reactive oxygen species, the lipid peroxides, all of that stuff is going to come as an inevitable consequence of this oxygen that you're distributing to your cells.
Yeah.
And so, we mentioned this before, but let's just sort of say it again for people and put a bow on this from an evolutionary perspective, right? So a very, very, very long time ago, there wasn't really much oxygen in the earth, but there was living things. And then oxygen comes on the scene, and we'll just call it the oxygen revolution. You basically say that after this oxygen revolution, many, many millions of years ago, power-hungry nervous systems appeared after that. And with them, apparently, the need for sleep. And then, although sleep since acquired additional functions like synaptic homeostasis and memory related stuff, the power law that we just talked about relating to daily sleep need and mass specific O2 consumption suggests that like this is the ancient purpose that evolved first. That had to do with the use of oxygen that was only enabled once there was oxygen on the earth. And you have this need because inevitably, so you're getting all of this ATP from the mitochondria. That's what you get. But inevitably, you're producing these compounds that lead to cell dysfunction, and you need to be able to compensate for those. And sleep is apparently serving that role.
That's at least the way I see it. That's my prejudice. I mean, it's a not entirely unfounded prejudice anymore, but yes, that's how I think about the problem.
Mm-hmm. And then, so you're not saying sleep isn't also for memory consolidation, these other things, but those are basically they stack on top of this one because they're all downstream of this energy metabolism part of the equation here.
Or they could even be independent of it.
Yeah.
If evolution just managed to take advantage of the off periods that were required for metabolic reasons, right? I mean, it doesn't mean that the other functions of sleep are directly downstream of metabolism.
Mm-hmm, mm-hmm. One of the things that you note is, including in humans, a common symptom of mitochondrial diseases of various kinds is this overwhelming sense of tiredness. And it's actually independent of, muscle fatigue and, moving around that day. And that would make sense because if you can't officially use your mitochondria to make cellular ATP, you're naturally going to feel fatigued.
Now, the interesting thing about these mitochondrial diseases is people have characterized these patients very, very carefully, and they tend to have normal ATP levels. They also have in many cases no problems with strength, with muscle, but they still have this overpowering sense of tiredness. And the way I think about it is, so many of these patients have mutations in the mitochondrial genome. So they have difficulty aligning the subunits of the electron transport chain precisely. So you could imagine, we talked before about the wire through which you pass the electricity, right? So these patients probably have a more resistive wire, an electron transport chain that opposes electron flow with a larger resistance. So what you need to do to get the same amount of ATP synthesis, you sort of need to increase the voltage, the driving force with which you pass the electrons through that transport chain. So you need a higher supply of electrons, and there we are again. It's the balance between electron supply and demand that determines the size of the electron leak. So my prediction would be that many of these patients have an increased electron leak, and that is the cause for their sense of tiredness.
Interesting. That makes logical sense to me. One thing I want to call out here too is it sounds like how well matched things are in this wire in the mitochondria dictates how readily the current flows and how nicely all the pieces fit together and how efficient our energy metabolism is. We normally think about mitochondria in a maternal centric way because we all inherit our mitochondria only from our mother. But it sounded like earlier you said some of the subunits of the machinery in the mitochondria are encoded in the nuclear genome. And so the nuclear and the mitochondrial genome both encode stuff that needs to be put together inside the actual mitochondria. And that would imply that both your mom and your dad sort of need to have pieces that match up well to have a nice system here with all this, that allows for good current flow and all of that stuff. So it's not strictly that mitochondrial function is entirely dictated by your mother, even though we inherit our mitochondria from the maternal side of our lineage.
That's absolutely correct. So the mitochondria, they are descendants from bacteria that were engulfed by another bacterium. And over time, they have delegated much of their genome to the nucleus. So the vast majority of mitochondrial proteins are encoded in the nucleus. There's a sophisticated machinery that imports these nuclear encoded gene products back into the mitochondria. But a small number and these are exclusively electron transport chain components, have remained resident in the mitochondrion. And it's an interesting question why. Why have the mitochondria not outsourced these 13 proteins? I've looked in the literature at some of the answers that people have proposed, and the one that sort of strikes me as as probably the most plausible is that what the mitochondrial genome gives you is local organelle restricted control, right? If you have a nuclear encoded mitochondrial protein and you get a signal that says make more of that protein, you don't know which mitochondrion actually requires the protein of the many hundreds to thousands in a large cell, mitochondria, right? So where do you target the gene product? Whereas if you make the stuff locally, you can sort of fine tune your electron transport chain depending on local demand.
Yeah, you can fine tune things more because you don't have to wait for protein subunits to ship in from further away.
Yeah. And you can produce what you need only where you need it.
Yeah, on demand. So you get this sort of nice, there's a spatial and a temporal specificity there. If I want to make a little bit more of this subunit, but only in the mitochondria, like over in this part of the cell, you can do that.
Yep.
And so this is a little bit of a different topic, but it's worth calling out. So people might, so the name for the idea that mitochondria used to be free-living bacteria that got engulfed by another one, that was Lynn Margulis's idea called the endosymbiotic hypothesis or endosymbiotic theory. And this is not purely just a theory. It's the thing that you mentioned about the nuclear genome containing genes that used to be in the mitochondria, that's actually a key piece of evidence here that basically the way this works, as I understand it, is the mitochondria used to be bacteria. And so their genes look bacteria-like compared to eukaryotic genes like the one in our nuclear genome, except you can actually see what are essentially bacteria-like genes in the nuclear genome. And that's actually the signature of what you mentioned, which is that over time, a lot of those mitochondrial, basically bacterial genes, integrated into the nuclear genome. And so we can actually see sort of a genomic record of this integration, this very ancient integration.
I would say that's probably the case, but I'm by no means an expert in sort of comparative in genomic archaeology and tracing evolutionary history. So
yeah. One person I've had on the podcast that I'll point people to is Michael Lynch. So I did an episode with him about evolution stuff. I would recommend that if people are interested in it. I want to talk to you a little bit about sort of what comes next in your lab with this story about cell metabolism and sleep and how all of the things work out mechanistically here. What are you guys building on from this work and from the lipid peroxidation sensor work to take the next steps?
Yeah. So there's, I think, three really pressing questions that we are desperate to answer. The first one is we've focused on these 30 or so sleep control neurons. A vanishingly small minority in the brain. And that's true for most animals. So for instance, C. elegans has a sleep control circuit too. Out of its 300 something neurons, there's only one cell that is responsible for sleep induction. In our brains, it's probably a few tens of thousands of cells in the hypothalamus that do the same job. So we think that these sleep-inducing cells measure the state of their own mitochondria as sort of a representation of what's going on in the rest of the brain. They are sort of like, to stay with an electrical engineering analogy, they're like circuit breakers or fuses. They see how well or how poorly their own mitochondria are doing, and then blow the fuse before it gets too bad for the rest of the brain. So we are trying to test that hypothesis by changing the fuse in the brains of flies. So you could imagine that if you have a circuit in your house that's rated for a current of 2 ampere, and then you put a 10 ampere fuse in there, you run into problems. You might set your house on fire. Mm-hmm. So what we are trying to do is equip the brains of flies with fuses that are overrated for the load that the brain is built for, and we will then try and investigate the consequences of chronic sleep deprivation, or look for the consequences of chronic sleep deprivations in the brains of these animals. And these consequences would be cognitive impairment, so learning and memory deficits, and also a shortened lifespan. So I think this is one pressing question, to understand what the relationship is between the sleep control cells and what's going on in the rest of the brain. Second, and we've touched on that already, is does the same mechanism operate in different species? Is this true across species? Especially, is it true also in mammals? And the third question, and we've also briefly touched on that, I think we are getting a pretty decent sense of what the cause of sleep is. It's the mitochondrial electron leak, we think, but we don't know what the function of sleep is. We don't really know what sleep does, what happens during sleep. Does a sleeping brain have a smaller electron leak? Is it simply that the leak gets plugged somehow? And if it gets plugged, how?
Yeah.
Or is there some active repair processes that are being initiated while you take your brain offline? So that's another really, really big, important question to answer.
Yeah. And, another thing I wanted to ask you about here is, we're talking about neurons in the brain, and a couple of things occur to me. So one, neurons, in a sense, neurons are sort of uniquely or especially sensitive to oxidative stress, in the sense that you can't replace most neurons in the brain. They're not a dividing cell, so they really need to track oxidative stress, because you can't just make a new neuron in whatever the mushroom body of the fly when you need it. You can't just replace a key circuit in the hypothalamus of a mouse or a human if those cells die from oxidative stress. The other thing is that I know this is true at least in vertebrates. Maybe it's true in flies as well. I know that basically the lipid membrane composition of neurons tends to be more enriched than many other cells in things like polyunsaturated fatty acids. So that's another way that neurons might be sort of extra sensitive to oxidative stress, and why they would really need to have mechanisms to track all of the things that we've talked about. What can you tell me there in terms of how uniquely sensitive to oxidative stress neurons are, and what that has to do with things like the fatty acid composition of the membrane and what the animal's eating, in terms of like if it's eating a lot of polyunsaturated fatty acids?
So I think that neurons have sort of three strikes against them that makes them especially vulnerable. And you've mentioned two of them. One is that they are not renewable. Two is that they have a lot of lipids, not just in terms of composition, that they tend to have lipids that are highly polyunsaturated, but they also have a lot of lipid. They have very extensive membrane systems, so their surface to volume ratio is very, very large, because they have these vast extended processes. I mean, so there's a lot of membrane that can get damaged in neurons. And the third strike, of course, is the large amount of oxygen consumption. So in humans, the brain accounts for 2% of body mass, but consumes 20% of the oxygen we breathe. And that, of course, also predisposes you to the risks of oxygen use, right? How the degree of polyunsaturation of your lipid membranes is controlled through diet, I don't know. I mean, there's clearly some influence, because some of these polyunsaturated fatty acids are essential, so you have to have them in your diet, and what you take in will certainly influence your lipid membrane composition to some extent. But there's, of course, also biosynthetic pathways that allow you to modify, as long as you have enough of these essential components, the repertoire of lipids in your cells according to what they need. But I think one dietary variable that certainly matters is simply the total amount of calories that you consume. There's a large body of evidence, again, ranging from flies to humans, that caloric restriction, so using your dietary intake of calories, reduces sleep need quite substantially. So
that makes sense, right? The less food components you're consuming that are going to funnel through this electron transport chain, the less or the less leak you're going to get overall, and therefore the less reactive oxygen species and lipid peroxides and stuff on average that you'll tend to form.
It would seem to be the case. Yeah.
Yeah. Interesting. Is there anything that you want to reiterate or any final thoughts you want to leave people with about this body of work that you do? This is, of course, not all that you've done in your career, certainly, but when it comes to sleep and metabolism and this sort of deep connection, what do you want people to take away from this?
I think we've had a very wide-ranging conversation, and it's been thoroughly enjoyable. I hope it's been as enjoyable for the audience as it has been for me. I think the key message is that that sleep is at its root a biochemical, the consequence of a biochemical problem that seems specific to the brain, and that surprisingly the brain has found no way to solve but to make us unconscious.
Professor Miesenböck, thank you very much for your time, for joining me across the pond there. This was fascinating. I'm going to put a link to one of these papers in the episode description. I'll also call out, so you mentioned Jeffrey West's work earlier. I've had him on a very early episode of the podcast, and I recommend his work as well. I think that if you listen to that one and this one back to back, they go together actually in a very interesting way and help explain how some of these things work. He studied sort of the way that body size and metabolism scale with these networks in biological animals. So fascinating stuff. I look forward to following your work on this stuff. This work that I'll link to, I think this is very beautiful work. I'll just tell the listeners, it's very hardcore. So if you want to go read these papers, they're very, very hardcore, but very well done. They span neurobiology and biophysics and biochemistry. So thank you very much for your time.
Thank you.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.